everyone, I am Sutapa, present in front of you to explain the next topic. Now I shall discuss about the cell division. It's a long topic. In the single video it will not be completed. So I am giving the part 1, part 2 like that. The cell division. Now I am discussing about the cell division 1. Cell division is the type which you know from the very lower classes. You have studied about the cell division, but in class 12 or 11, that is in 11, 12, in competitive exam also and in board also, you have to know the cell division in details. Now, what is the cell division? That is the reproduction of the cell. That is, reproduction means it will form from two kinds of its own type. The cell will, we know that the cell will form the two types of its own kind, but there are many types of the cell division like that. Number 1 is that mitosis. Number 2 is mitosis, and number 3 is meiosis. All the criteria are different in different cases, and mitosis is the type which we can see in case of the lower organisms, either lower plant or lower animal. And this is the direct cell division. ये डायरेक्ट सेल डिविजन क्यों कहा जाता है? क्योंकि न्यूक्लियस जो डिवाइड होगा उसको that it will be divided in approximately same size, not the exactly same size. Now, what is the content of the nucleus? Nucleus में क्या रहते हैं? Generally chromosomes रहते हैं. तो chromosome should be divided in such a manner that it will be divided not exactly but apparently the same number. Like suppose I am giving you the diagram like this. It's a bacterial cell. It has a nucleus in this way. In mitosis, mitosis में क्या होगा? Mitosis में इससे दो अलग-अलग सेल होंगे. Mitosis में also, in a case of mitosis also, it will produce two cells, but in that way. Suppose they will be divided in that way. So they have the nuclear material which is almost same, but not the exactly. Suppose घर में मम्मी एक सेब को दो बच्चों को दे रहा है, तो बीच से नाइफ देके कटी है, तो it can't be, if you count the cells, the both the cell numbers can't be same, can't be just same, but they know that we are getting the same amount. So this is the unscientific method almost by which we can divide anything into two divisions. But in case of the mitosis, which is mitosis and meiosis, both are present in higher organisms. And these are scientific method to separate the cells. Why? Because in mitosis and meiosis, we can see that the spindle fiber is formed. Spindle fiber, by the formation of the spindle fiber, the chromosomes, they get aligned in this manner. This is the spindle fiber, suppose. So chromosomes are arranged in this manner. Suppose it has 4 chromosomes. One will come to here and another here. One will come to here and another here. So naturally, there is the exact division of the chromosome. This is the main difference of mitosis with the mitosis and meiosis. Now, what is mitosis? In short, we can say that the mitosis is the equational division. What is equational division? Equational means that the daughter's mother cell and the daughter cell will be of same kind, exactly same kind, and their chromosome number will be also same. Suppose a diploid cell of our body cell, our autosomal cell or diploid cell of us, that will be divided by mitosis, it will form two twin cell. Both the cells are twin. So exactly equational division is the same, एकदम पूरा दो सेम काइंड का बच्चे होंगे सेल का, ठीक है. तो और इन केस ऑफ द मियोसिस इट्स नॉट दैट, because meiosis is divided into two, that is the meiosis 1 and meiosis 2. Now in meiosis 1 it's the reduction division. Why reduction division? Because if the diploid cell is divided by meiosis 1, then the diploid cell will form two haploid cells. So two haploid cells are formed, and then meiosis 2. By meiosis 2, those two haploid cells will form another 2 to 4 haploid cells. So meiosis 1 is the reduction division and meiosis 2 is the equational division, just like the mitosis. So this is the reduction division and this is equational division. This is the very basic idea about the cell division. Now I am coming to the point that why the cells are getting divided. In case of mitosis, if we consider, suppose cell has nucleus and cytoplasm and that has the proper ratio. Suppose 3:1 or 4:1, in that way they have the particular ratio. If the ratio gets changed, that's known as the karyoplasmic index number 1 due to the why. Just to think it's in very easy way that in a family they have two children, parents and two children. Now in a small home, or they could easily accommodate themselves and happily used to stay. Now इन दोनों का शादी हो गया और दोनों का दो दो बच्चे हो गए. Now their accommodation is, there will be accommodation shortage, isn't it? So now the nucleus of the family is bigger than the cytoplasm. That means घर जितना बड़ा है उसके हिसाब से उनके members ज़्यादा हो गए. So nucleus is increasing and cytoplasm is, in comparison to that, it's not increasing. Cytoplasm is remaining same. So naturally there will be the space shortage, there will be the shortage of many things, and then the parent will decide that they should make two different homes for the two children. And this is the way like that in case of the mitosis also. So number 1 is change of the karyoplasmic index. Karyon means nucleus, plasmic means cytoplasm here. Ratio between cytoplasm and nucleus. Now it can be expressed by Ki, karyoplasmic index is Ki by Vn minus Vc. Now Vn, what is Vn? Volume of the nucleus. And Vc is the volume of cytoplasm. And next is surface volume ratio. Number 2 is surface volume ratio. It's changed in the dividing cell or the cell which are going to be divided. There the ratio of the surface volume ratio will be changed in such a manner that the volume of the nucleus will be more than the surface. Volume increases than surface. And then also the cell divide. So this is the main motive of the cell division, why cell division is taking place, and what's the main target of it, to produce its own kind. This is about the mitosis. And then we are coming to the, in mitosis or any cell division, there are two parts, that is suppose from one division to other division. If we go, then the events are taking place in cyclic manner. That's forming the cell cycle. So what is cell cycle? It's the sequence of events which take place in between two cell divisions. And this cell cycle is divided into broadly in two divisions. Number 1 is interphase and number 2 is M phase or divisional phase. M from mitosis. Now, what is interphase? Interphase initially it was known, पहले ये कहा जाता था जो interphase is the resting phase. As why it's resting phase? As no activities, no modifications, no alterations, no changes are observed from outside in this phase. So it's known as interphase is resting phase according to the scientists. But now it's observed that the interphase is highly metabolic one, as it's taking preparation to enter in the divisional phase. ये जस्ट ऐसा है जो कोई बच्चा घर में पढ़ रहा है लेकिन वो किसी को पता नहीं है जो क्या कर रहा है, पढ़ रहा है क्या, टीवी देख रहा है और क्या नेट लेके बिजी है, कुछ नहीं पता. That girl or boy is taking preparation for the competitive exam or any tough exam or for board exam also, but the outsiders can't understand that. Outsiders, they have no idea about this, as they are thinking that she or he is remaining inactive. But after the competitive exam, it's revealed that that student was preparing himself or herself for the to appear in the very tough exam. Like that, in interphase, the cell is taking preparation to enter in the divisional phase, but it has no such symptoms which is observed from the outside. Now, in interphase, what are the subphases? The main thing is that interphase is divided into 3 subphases. Number 1 is G1 or gap 1 phase. Number 2 is S phase or synthetic phase. And number 3 is G2 phase or gap 2 phase. Now, what's the main target of the G1 phase? G1 phase is the first phase of the interphase in which many replications of the organelles are taking place, as well as different proteins which are required in the next phase. Next phase is very important, why I am telling later. And in this phase, all these required things are replicated or produced, like the different organelles. Suppose घर डिवाइड करने के लिए दोनों फैमिली का पूरा फर्नीचर्स चाहिए, पूरा यूटेंसिल्स चाहिए, गैस ओवन चाहिए, एवरीथिंग दे नीड the replicated one, that means the duplicate copy of each other. Now in this case also, the cell is dividing, that means mitochondria should be divided in the two cells equally, Golgi body should be divided in the two cells equally. In this way the organelles will be divided equally, so the replication or formation of the organelles are taking place in G1. And another thing is that the S phase is the synthetic phase. S phase is very important, as here the replication of the DNA takes place. DNA, we know that the main thing of the chromosome, nucleus, and nucleus will be divided, so chromosomes will also be divided, and if we break the chromosome, ultimately we can get the DNA. So DNA should be replicated, and replication of DNA is the most important thing, and that will take place in case of the S phase or synthetic phase. So another responsibility of the G1, that it has to complete the formation of the protein, histone protein, and the other proteins which are required for DNA replication. Now the replication of the DNA, the nature of the replication of DNA, it should be in this way, that is the semi-conservative method. In class 12, you should study about this. What is semi-conservative? Just I am giving an idea about it. That during replication of the DNA, the mother DNA is uncoiled or unwinded to form the two strands. Now each strand will form the new one. With the blue one, the red one is the new. Now what is semi-conservative? That's in semi-conservative replication, this condition will retain. That means in a new molecule there should be a new strand as well as the old strand. So partially conserving the parental structure. That's why it's known as the semi-conservative method. So DNA replication takes place in S phase. This is the main thing of the S phase, and after that the G2 phase, where the other things are replicated, and it's just completing. It's just exam ke pehle last preparation. G2 is the last preparation to enter in the divisional phase. Appearing in the exam, before the day, you just preparing yourself completely. That is the G2 phase, the gap 2 phase. Now another thing I am telling you, that in case of the G1, after G1 phase, some cells they go to the an inactive phase, that is the G0 phase. Now this G0 phase is like that. Here the cell will remain in inactive condition. It will not divide. Remain very in idle manner. Like in case of the neurons, we know there is no division. Some liver cell, some heart cell, they don't divide ever, so they stay in the G0 phase. But if they need, then they can come to the S phase and G2 phase, can complete their division. But if any cell enter inside the S phase, then after that it has no way to back. अगर इस फेज में आ गए तो इसको कंप्लीट करना है, पूरा डिवीजन कंप्लीट करना है, उसके पहले ये छोड़ नहीं सकते हैं. सो दिस इज द थिंग दैट if the cell enters within the G2 phase, S phase, then it has to complete the total interphase, and after that divisional phase. Now that's all today. In the next video I shall explain other things in very details. Bye for today.